Welcome to my lecture. I am Dr.M.Isai from Department of Zoology. Now I am going to explain about the instrumentation and procedure of AGAROSE gel electrophoresis. Before going into detail, I would like to give a brief introduction about electrophoresis. What is electrophoresis? It is the process of separating and characterizing the molecules based on their size and its charges. It can be defined as a process to identify, separate, and characterize the biological molecules such as DNA, RNA, and protein. What are the aims and objectives of gel electrophoresis? We can identify the biomolecules like DNA, RNA, and protein from the biological samples. We can determine the size of the target biomolecules. We can run biomolecules under electrical current and also to estimate the size of biological molecules of our target sample. What are the different types of electrophoresis? Electrophoresis is of two types. Number one is vertical electrophoresis. The second one is horizontal gel electrophoresis. In vertical gel electrophoresis, we can run the samples from upside to downside, and this process is discontinuous because in the system we are using two different types of gel. One is stacking gel, another one is separating gel. In stacking gel, the protein molecules are arranged as a stack, then it's moved to the separating gel. There the protein molecules are separated based on their molecular weight. This vertical gel electrophoresis is commonly used in proteins separation. For the separation, we are using polyacrylamide as a matrix. Hence, this type of vertical gel electrophoresis is also known as polyacrylamide gel electrophoresis or PAGE. The second gel electrophoresis is a horizontal gel electrophoresis. Here, the samples are run continuously. It is parallel to the surface, then DNA get separated. Here we are using agarose gel as a matrix. Hence, this type of horizontal gel electrophoresis is also known as agarose gel electrophoresis. Here in this session, I would like to give a detailed explanation about the agarose gel electrophoresis. It is a method of gel electrophoresis used in biochemistry, molecular biology, genetics, and almost all biological sciences, and also in clinical chemistry, used to separate a mixed population of macromolecules such as DNA and RNA in a matrix of agarose. Agarose is nothing but it is a natural linear polymer. It is extracted from seaweed that forms a gel matrix by hydrogen bonding when it is heated with a buffer and allowed to cool. They are the most popular medium for the separation of moderate and large size nucleic acid and have a wide range of separation. What is the basic principle involved in agarose gel electrophoresis? When a potential difference is applied across the electrodes of electrical tank, it's containing agarose gel, and the biomolecules are loaded in the well of this agarose gel. Then they get separated according to their molecular size and move to their respective electrodes. Here the bigger molecular weight have more molecular size and the smaller molecule have small molecular size. Here the agarose gel acts as a sieve. As in the sieve, large particles stay above and the particles which are smaller than the pore size passes through it. Similarly, the larger and the bulky molecules stay behind, whereas the small molecules move faster and quickly towards their respective electrodes. Generally, DNA is negatively charged. When placed in an electrical field, DNA will migrate towards positive electrode. That is, usually positive electrodes are in red color. This process may be imagined like running competition. The one who is thin and flexible, will reach the destination easier and very soon when compared to fat and bulky person. The following are the equipments required for the conduction of agarose gel electrophoresis. Number 1 is electrophoretic chamber and power supply. Number 2, gel casting tray, which is available in variable size. It's composed of UV transparent plastic. The both end of the tray will be covered with transparent tape at the time of gel casting. Then it removed at the time of electrophoresis. And third one is sample comb. The sample comb used to make a belt in the gel. And the fourth one is electrophoretic buffer. Usually electrophoretic buffers are tris acetate EDTA or tris borate EDTA. It is commonly called TAE buffer or TBE buffer. Then loading buffer or tracking dye. It contains xylene xylol, bromophenol blue, glycerol that allow the sample to fall into the sample well. The next one is staining. DNA can easily visualized with the help of ultraviolet lamp when electrophoresed in the presence of ethidium bromide. So the staining is ethidium bromide. And coming to the transilluminator is used to visualize the stained DNA. It is transilluminator is a ultraviolet light box. Here in this picture shows the setup of agarose gel electrophoresis. The yellow one is electrophoretic tank, agarose electrophoretic tank. The blue one is a comb. Comb is used to make a well, and this one is a tray, gel casting tray. Then electrodes. Red one is connected with the power supply, and the black one is connected with black end of power supply. And coming to the steps involved in agarose gel electrophoresis. In agarose gel electrophoresis, we should concentrate on the percentage of gel because the percentage of agarose is based on the size of the DNA fragments. Normally, they range from 0.2 to 3% agarose gel. First, we have to prepare the gel. Agarose powder mixed with electrophoretic buffer and heated in a microwave oven to melt it. Then we have to add ethidium bromide. This ethidium bromide is used to visualize the DNA. After cooling the solution, it is poured into the casting tray containing a sample comb. It's allowed to solidify. After solidification, we should remove the comb carefully. After removing the comb, it is ready to inserted into the electrophoretic chamber and is covered with buffer. Then we have to prepare the sample containing DNA along with the loading dye or tracking dye. This should be pipetted into the sample well. Then cover the electrophoretic chamber with the lid. Then supply the current. For applying the current, we should concentrate on two different voltage. One is a 50 voltage, another one is 100 voltage. Initially, we should give 50 voltage. Once it's come out, sample come out from the well, we can turn it into 100 voltage. Then how can we check the current flow? We can check the current flow with the help of observing bubbles comes out from the electrodes. Then DNA will migrate towards the positive electrode. As I said earlier, DNA is a negatively charged particles. When placed in an electrical field, DNA will migrate towards positive electrode. The DNA migration in the gel can be judged by visual monitoring with the help of tracking dye. And coming to the technical setup of agarose gel electrophoresis, in a gel casting tray containing a solidified agarose gel after removal of the comb, then loading this DNA sample into the well in the gel. After loading, we should pour the electrophoretic buffer, then cover the tank with the help of lid, then connect the positive and negative electrodes to the power pack. We can check the current flow with the help of observing the bubbles comes from the electrode. Then this, we can switch off it and remove from the gel casting tray, and place under the UV transilluminator. Under transilluminator, we can see achromatic DNA stained bands. Coming to applications, agarose gel electrophoresis is widely used in the estimation of the size of DNA molecules and also the product of PCR analysis. After running the polymerase chain reaction, we should assess the amplified DNA with the help of agarose gel electrophoresis. And also used in northern and southern blotting. Before running the northern and southern blotting, we should separate the DNA or RNA molecule for the further analysis. And we can estimate the unknown DNA molecules using DNA marker or DNA ladder, which contain DNA fragments of various known sizes. We can compare these known sizes with our unknown target DNA molecules. And also, rough estimation of DNA quantity and quality can be assessed with the help of agarose gel electrophoresis. Thank you.